Hello everyone, I hope you're all doing well after this first week of the quarter, and I hope that you found MCDB 134 or Virology interesting. I know I have. And yeah, there's been a lot of material that's been covered, and as I mentioned in my first ever video, this is a way for me to reveal, and I hope that it will, if you're watching this, there will be some sort of benefit that you could derive from this video as well. And yeah, I'm going to try and keep these videos as short as possible, definitely less than a lecture length. And you know what, let's just get started. Okay, so this first week we've learned a couple things about viruses. We've learned about, Dr. Samuels has taught us about the history behind virology, the structure of viruses, their classification, or how to classify the viruses, the methods that we use for classification, assay techniques, and the measurements that are used in the assays. So with the history, it's really simple. From what I could gather from lecture, there were 6 people who contributed, who greatly contributed to the field of virology, and that's Ivanovsky, Bagerink, Lofer and Frosch, Reed, Bong, Tort, and Day. Now, names don't mean anything, so I'm gonna go through how each of them contributed to the field of virology. I believe in historical order. So Ivanovsky discovered viruses when he found that there was a filterable disease agent that it went past the filters that bacteria can pass. So he found out that viruses were filterable. You know, they were particulars. You could take them out. They weren't just some weird life force, so to speak. I do not know. Sorry about that. The next person after Ivanovsky is Bagerink, and he discovered that viruses are able to multiply. Now, don't get this confused with viruses being able to divide. Viruses do not divide. They can't synthesize their own proteins. They don't use their own energy. But they are able to multiply, and they do that by entering cells in life that can use energy and can make proteins. Now the now these next two, they worked together, I believe, and their names is Lofer and Frosch, and they discovered that viruses, that there were animal viruses, and I believe I can't remember. I believe they discovered that there were animal viruses by looking at, let me see here, by looking at foot and mouth disease. I think that's like a cattle virus or something. So yeah, they discovered that there were animal viruses. The next person is Reed. I'm guessing he was looking at yellow fever, and he discovered there were also, in addition to animal viruses, there were human viruses. Bong found out that there are viruses that cause cancer, such as HPV or hepatitis. And finally, Tort and Day discovered the bacteriophage. So we'll go back to this. So Dr. Samuels gave us a brief history on the field of virology. From there, Dr. Samuels started talking about the structure, and some of the things that from my notes is Dr. Samuels said that viruses are small, they multiply but don't divide. And going back to the fact that they're small, they're 20 to 300 nanometers, and they are made of, and they contain RNA or DNA. Now, here's the important thing. Viruses don't have both RNA and DNA. Like human and bacterial cells do. They either have one or the other. So with that, viruses can be can have either RNA or DNA. If they are a DNA virus, then they can be either single-stranded or double-stranded, and you just end there. For RNA viruses, it's a bit more just slightly more complicated. So RNA viruses are single-stranded and double-stranded. Within the single-stranded RNA category, there are positive single-stranded RNA viruses and negative single-stranded RNA viruses. Now the positive means that the RNA of the virus is in a sense, I guess, meaning that if that virus injected its positive sense RNA, its RNA can be transcribed and translated immediately into proteins. Whereas if it was a negative, or I think it's or missense, or I forget. Basically, it's the reverse of a positive single-stranded RNA virus, where if it injects a single-stranded RNA, it can't immediately have its RNA transcribed and translated. It has to go through some more steps before its RNA can actually give rise to proteins. So there's that. Let me go to my notes and show you guys. Oh, it's not there. Okay, my bad. Sorry, this is my second video. It's a bit nerve-wracking. Next few things about the structure of viruses is that they have, and some viruses have an envelope. So that means that they have a sort of like a lipid bilayer surrounding them to give them extra some form of protection or another form of entry into a cell. And a virus can multiply even if its host cell is not dividing. So when I hear this, I think of, chickenpox or polio. Our neurons aren't dividing, and yet those two viruses are still able to enter our neurons and somehow multiply. And that's probably pretty simple. It's just the fact that they don't necessarily have to, the reason why they're able to do that is because they just simply commandeer the protein synthesis and DNA synthesis mechanisms of our neurons. Our neurons still they don't divide, so their DNA mechanisms are dormant before infection, I guess. But at the time of infection, then as I was saying, our neurons, even if they're not dividing, still need to make proteins, for example, like acetylcholine. Well, when polio comes in, it takes all the infrastructure to make acetylcholine and decides to trick our neuron to making a bunch of the poliovirus instead. And then it takes up the neuron's DNA, kills a neuron, and steals our neuron's DNA and turns it into more poliovirus genome. So that's why, viruses don't necessarily need a dividing cell to multiply. And then Dr. Samuels also talked about the symmetry of the virus. So there's icosahedral symmetry, helical symmetry, and bullet-shaped viruses and pox-shaped viruses. Now, so with like the basic structure and characteristics of viruses established, Dr. Samuels transitioned to classification. So he said that there used to be an old method of classification, and that old method was done by pathology, but nowadays we do it by biochemical and physical properties. Now, before I go into more about classification, I actually need to jump back to structure. So I'm pretty sure everyone has seen pictures of viruses in textbook somewhere. I'm assuming that if you made it this far as a bio student and specifically a microbiology student, you've basically know what a virus looks like. But just in case, if maybe you're taking this as an elective or whatever the reason that you don't know, I have on screen a picture of just on the internet, a picture of a virus, and I'm going to show you later drawing. So you can see all these captions. Here's a virus. So this is one of the structures of a virus, and you can see that in here, that little coil right there, that's its genome. This particular one is DNA, because you can see that, or well, it could be RNA. It's definitely double-stranded though. That genome is protected by a protein coat, which we call a capsid. Yeah, so you can call it protein coat, but for this class we call it capsid. That capsid is made of these things called capsomeres. Now this capsid, some viruses have it and others don't, but in this particular picture they have this virus capsid inside of another shell, which is basically a membrane. I think it's a lipid bilayer because it is derived from the cells. It's derived from host cell membranes, but you should ask Dr. Samuels about that, and I will probably ask him too, like what this is specifically made of. I don't think you really need to know what it is exactly made of, but just know that some viruses have a envelope surrounding them and others don't. And viruses have oh wait, I need to show you my drawing. So this is my drawing. Pretty shitty drawing, but it got pretty much all the points that Dr. Samuels had in class. So you can see nucleic acid right here, capsid. Now the core is just basically this whole thing right here, just the capsid and the genome. That's called the core. But basically the core has a nucleic acid, and there's a capsid right here, this hexagon thing, and then that capsid has these rectangles called capsomeres, which basically make up the capsid. And then I wrote in big letters right here, optional. This virus could, some viruses have this envelope, others don't. And yeah. So now that we have structure out of the way, again, like I said before, Dr. Samuels, after he had established very basic structure and some of the characteristics of viruses, he went into their classification. So viruses used to be classified by the disease they cause, but nowadays we use biochemical and physical properties, of which the biochemical and physical properties, they're the ones that we just mentioned in the structure and characteristics. Those are some of the ways that we can classify viruses. We can classify them by either whether or not they are RNA or DNA, or single-stranded RNA or double-stranded RNA, or positive single-stranded RNA or negative-stranded RNA, or if it's DNA virus, then if it's single-strand DNA or double-stranded DNA, or you could also, instead of classifying them by what disease they cause, you can classify them by whether or not they have an envelope or not. So many things. There's a lot, and I don't think Dr. Samuels went into detail the different, I'm sure there's so many characteristics about viruses that he didn't even cover. Now, with that said, we have to know the methods, some of the methods that scientists use to classify viruses. One of those methods, so there are 4 methods. The first method is membrane filtration. Let me pull up the notes. So membrane filtration. And in this method, you basically use basically a bunch of sheets of bunch of filters, and each of those filters have different pore sizes, and you basically stack them up from the top to the bottom. You have filters that have big pores, and on the bottom you have filters that have small pores. Now, the thing about this is you know what the pore size is. So let's say the top pore size is, the top filter has pore sizes of 300 nanometers, and the bottom filter has pore sizes of 20 nanometers. Then when you are filtering out viruses, you will capture all the viruses that are 300 nanometers big will be captured on the first layer, and that's and now you can classify that virus. But if it's smaller than 300 nanometers, like let's say if it's 20 nanometers, then it will be captured on a bottommost filter, which has 20 nanometer pores. So that's one way of that's one of the methods scientists use to classify viruses, is by membrane filtration. The next method is to use ultracentrifugation. Now, this is, you will basically separate the viruses based on their density. So you centrifuge the viruses in a medium like sucrose, or I'm guessing like like a sugar medium, and it doesn't have to be sucrose, it can be a bunch of other stuff. I don't know what those things are, I'm not in the lab, and Dr. Sanders didn't go over it, but just to get the idea across, you can centrifuge on something like sucrose. And then when you're done centrifuging, you'll get like different, if you have like a mixture of different viruses, those viruses will be centrifuged out to certain portions of the sucrose. And that sucrose is, you also, the way that you have the sucrose set up, like when you put the sucrose in a centrifuge tube, the top portion of the centrifuge tube might have high density sucrose, and the lower portion of the tube might have a lower density of the sucrose. And so when you centrifuge a virus, it will go from, the ones that are, you get different grades of the virus, and where they are relative to the sucrose, you can infer the density. I will try to show, I think I drew a picture at in my notebook, and I'll try to show that at the end of this video. It's pictures, picture's worth 1,000 words. Yeah, so I'm sorry I don't have a picture here, but it's very hard to draw using a mouse. But anyways, let's continue. So next, the third way that Dr. Samuels said scientists use to classify viruses is electron microscope negative staining. So basically, you put a negative stain on like your culture of viruses, and basically the virus will stain differently than its surrounding environment, and then you put that underneath an electron microscope, and then from the colors of the picture and so forth, you can determine its shape. And I'm guessing if you have a reference marker, somehow you can determine maybe its size too. But basically, if you want to know if a virus is icosahedral or helical or bullet-shaped or pox-shaped or bacteriophage, and this is one of the ways you can do it, EM negative staining. And finally, the fourth way is to use X-ray crystallography. Basically, it's the same method that Rosalind Franklin used to find the structure of DNA. So because viruses are so dang small and they're like only slightly bigger than DNA, you can use X-rays to find out more about their structure that way. And something that the book pointed out was when people found that you could use X-rays to learn more about viruses, that actually lended evidence that viruses live on the they're like on the edge of chemistry and biology, of chemicals and life. So it had a lot with X-ray crystallography. There was a lot of implications of perhaps the first or like the precursors of life could have been a viral-like particle and so forth. You can read in a textbook. I forget what page, but you can find somewhere in like chapter 1, I believe, of the textbook. So then, now the different. So I know this is a bit more related to. So we've already gone through the structure and all these characteristics. I know in my notes it's in a bit of a different order, and I reorganized it to make this video. So we'll skip all this. Oh, actually, there's this capsule mirror formula, which I think this may be covered on test. That's my gut feeling. So if Dr. Samuels ever asks, or like if you're in the lab and you ever need to know how many total capsule mirrors a virus is made of, then you use it by using this formula. N equals 10 times n minus 1 quantity squared plus 2, where capital N is a number of capsule mirrors, and N, you know what? I actually need to come back to this. Total number of capsule mirrors. I must have had a typo. Oh yes, yeah, capsule mirrors. Sorry. So capsule mirrors. So total number of capsule mirrors equals n, capital N equals 10 times n minus 1 quantity squared plus 2, where capital N is the total number of capsule mirrors a virus has, and lowercase n is the number of capsule mirrors per edge of the virus. So it's pretty complicated for me. I personally would go to Dr. Samuels or any of the TAs, or to clarify this formula and like what he means by edge and so forth. Maybe I'll put this in another video or something. Yeah, so all that. Now, so with classification, so Dr. Samuels was on Wednesday. He finished, I think it was a Wednesday. Yeah, Wednesday he finished methods of classification. Then Friday he talked about assay techniques. So with assay techniques, well, when you're studying viruses, using cultured cells is basically like the most basic. That's the simplest model to do research on viruses and so forth, because they're so simple. It's just a cluster of cells, put some viruses in, the cells get infected, you study them. If then you need to go to the next level complexity, like let's say if you're trying to create a vaccine or something, cultured cells might not be, your vaccine might work great with cultured cells, but now you need to know like how it would look like in an actual animal. So the next step of complexity is to use embryonic cells. Usually, as I have typed here, usually the embryonic cells come from chicken eggs, but in short, it's the next level of complexity after cultured cells is embryonic cells. And then after that, the most complex that you could go is to have an intact animal. Usually we use mice, but again, for vaccines, you might go from cultured cells to embryonic cells to mice and then to humans or something. And usually intact, as I have typed here, intact animals are used to elucidate the pathogenesis, study a bunch of stuff that I will probably never know in the future, but that's okay. Now, within cultured cells, there are three cultured cell types. There's a primary cultured cell, or primary cell culture. Then there's a strain cell culture, and then there's a continuous cell culture. In a primary cell culture, primary cells are just basically made from tissue of animals, and they only last about 10 generations of divisions. And so then you can use cell strains, which are derived from the primary cells that we from primary cells, and they last a bit longer, tenfold actually. So they last about 100 generations of division. And finally, you have continuous lines, cell lines. That's just, those cell lines are stuff like the HeLa cell lines, which have an infinite lifespan, and yeah, you could use those to study viruses. The only thing you need to be careful though is that with continuous lines of cell lines, I think Dr. Sanders was trying to say, like, you need to be careful that you basically have pure continuous cell lines, because those cell lines can undergo, because they live forever, they can undergo certain changes after some time. Like they could become cancerous or whatever, or they just might not work. They might stop working. He didn't go into much detail, but then there are the then Dr. Samuels went into some detail about the cell lines that are important for his class. On his handout, actually, he gives like a huge list of different cell lines that researchers use, and I'm guessing each one has a different purpose. Like you might use them to study one thing but not the other. But he said for when I was writing this down in lecture, there seemed to be 9 important cell lines, and you can see on my screen there's a HeLa cell line, which is a human cell line, and then there's the L cell line and the 3T3 cell line, which are I think they are mice cell lines. They are mice cell lines. The L and the 3T3 are mice cell lines. Then you have the CHO and the back, or CHO and BHK cell lines, which come from hamsters. Then CVI and BSC, which come, and a Viro, which comes from monkeys. And this one, HEC293, comes from a human cell line. And of course, when you're using cell lines like these, which I believe are all continuous, but he didn't say anything about that, so you can ask Dr. Sanders about that. But when you're using these cell lines, you need to, you also have to like culture them in different media types, and depending on your situation, you'll use different medias. But some of the medias that he went over for these, for culturing them, is minimal essential media, Dubeckel's medium, which is basically this, but twice the amount of nutrients, and then Jocklick's medium, which just he said that it was basically the minimum medium without the calcium, and you don't want the calcium there, so you could prevent clumping. And so scrolling down the notes, so towards the end of Friday lecture yesterday, he went over 3 different cultural layer types. I don't remember him saying anything about the significance of these culture types. He just said that these are 3 different cultures that you can have. You can have a monolayer culture, which is just, like on a petri dish, with the media, with one of these given medias, you just have one layer of animal cells, just one cell thick. The next one is the roller culture, which is basically you take like a tube or like a sterile jar or something, and you coat the inside with the media, and as you slowly spin it inside your incubator or something, the cells will start to form like a 1 cell layer thick culture around the whole inside of the jar. So you can imagine, like, this is, if you want a lot of cells in a small space to do your experiment or your research, you might want to use roller culture. I guess that's what you could use. And then suspension cultures, which is where the cells the host cells, or the cells that you infect with the virus, are just free floating in the medium. And then finally, assays. These are the measurements. So these are the measurements that are used in the assays. So in the beginning, there was the cytopathic effect, which was a crude assay, and basically that was Dr. Samuel says that it's histological evidence, but basically it's you have a culture cells, infect the virus, and see what happens to the cells. You know, do they shrink? Do they expand? Do they die? Do they multiply even faster? So, as I have written down here, these are the different viruses: Picorna, Pox, Rota, Rhabdo. If you infect a culture of cells with these viruses, then you will see shrinkage and death of the viruses. If you infect a culture of viruses with adenoviruses, you'll see like an aggregation. You'll see an aggregation of your culture cells, and then something that will detach off of the surface because they've been infected by the adenovirus. And then there's a Myxoviridae, which you see fusion of the cells. I don't know what he means by fusion though, but hey, apparently see fusion. Yeah. Again, this is why as I said in my first video, like my first ever video, go to lecture, read the book, ask the TAs for help. It may be tempting to think, oh, look, there's this guy, and he put some videos of MC134 on the internet. Well, I'll skip lecture and watch his stuff instead. No, don't do that. Please don't do that for your sake. Go to lecture, read the book. Just go freaking lecture. You're paying to be here. You're paying the professor to lecture. Just go to lecture, so that if you ever see something like fusion, you're like, what the heck is that? You can ask in lecture, right? And not have to like ask the YouTube video like, what's fusion or whatever. So anyways, going on. So after so one of the ways, a very crude way, very like, I guess, a simple way to look, a non-quantitative way to look at to detect if there has been a virus infection, to look for cytopathic effect, like what happens to your culture cells. I guess a slightly more sophisticated would be the plaque assay, or it's more quantitative rather than, it's not just qualitative like the cytopathic effect assay. The plaque assay is, its benefit is that you also get a quantitative measurement, and the way this works is, I know like for bacteria, when I did backpath lab last quarter, I remember doing like some of these plaque assays, or no, I was microbiology lab. We did some plaque assays, and what happened is like you take you grow some bacteria on an agar plate, and then you put like you put your bacteria on your agar plate, and then you also infect them immediately with your virus, and then you put like top agar on top of everything. So basically you have like a sandwich or hamburger of bacteria and virus. Top and bottom buns are the agar, and in the middle you have all this bacteria and virus mixing with each other, and the virus is killing the bacteria, and the bacteria is dividing, and so forth. And what happens is, after a few days, plaques form. So you'll see that there's a lot of you'll see like a whole lawn of bacteria, but inside that lawn there are like these circles, plaques, we call them, but like these circles are like just clearness. And the reason why it's clear is because the bacteria got killed by the virus. So with that, the theory is that you only need one virus to kill, you only need one virus to form this giant plaque in the middle of a bacterial lawn. So doing some math and so forth, you can find how the concentration of your the virus of your virus lysate that you put in and so forth. I think I'll do a separate video on that, but just know that the plaque assay is a quantitative way of measuring for viruses, detecting viruses. So yeah, that's pretty complicated. If you went to discussion section, you'll know exactly what I'm talking about. And then to finish our lecture, Dr. Samuels then had he literally said like 3 sentences about the transformation assay, but basically the virus can sometimes just change the way that the cell works. So I'm thinking something like hepatitis or HPV, like with hepatitis, you know, which is a disease that that affects 1/3 of the global population. You know, yes, some of the liver cells die, but a lot of the liver cells also live, and there's a bunch of viruses flowing around inside the liver cells, and those liver cells don't necessarily die. They can, but maybe not. But what it could also happen is they become cancerous from the hepatitis virus. So taking that into a lab setting, you know, if you're like growing a virus and there's no plaques forming, and you know you don't see any shrinkage or aggregation or fusion, you're like, oh, maybe the virus doesn't do anything. Well, think again. You can look for transformations instead. Like, you know, the cell becomes cancerous, and you're like, oh, it didn't die. The cells didn't die. The cells didn't form plaques or whatever, but it changed into something else that's not supposed to be. Perhaps the virus has something to do with that. So, and that was pretty much everything that was taught in class for the first week. Now, again, like, I'm going to say this in every single video, which is basically, don't just rely on this video. You know, you can see, you know, first of all, these are some of the first videos I'm doing. I'm very nervous. You know, I keep pausing and stopping. I'm not like an experienced lecturer or teacher like Dr. Samuels. But in addition, I just don't know as much stuff as he does. So it would greatly benefit you to go to lecture. You know, go to lecture, take notes. Don't sleep in class. You know, go to lecture or whatever you do, and then read the book. Go to discussion section, ask the TAs for help, ask Dr. Samuels after class or schedule appointments with him to get help with the material. You know, like, I'm going to do this too, but like if you see like the word fusion, like what the heck does fusion mean? You know, you can go to Dr. Samuels for that. But again, you know, I find making these videos very fun, actually. And this is actually a good way for me to, you know, I'm reviewing the material, so I'm having a lot of fun doing it. And I hope, you know, maybe some of the analogies I use or some of the words I've used, perhaps you're able to understand better. And I hope that this video has helped you. And yeah, I'll see you in the next video.